So, are you ready? Here we go with chapter number 1, sexual reproduction in flowering plants. What do you mean by flowering plants? They are simply the angiosperms, which means angiosperms are those plants which bear flowers in their lifetime. So, here we go. In this chapter, now you will be dealing with 5 topics, which is the first one, flower. You'll be learning the structure of flower. Then you have what happens before fertilization, that is pre-fertilization. The fertilization taking place in angiosperms, you call it as double fertilization. Then you have the events that take place after fertilization, which comes under post-fertilization, and some special cases and special examples of apomixis and polyembryony. These are the important topics which we'll be dealing with in this chapter. I'll skip the introduction part and going to the structure of a flower, very basic thing. What we have, the basic structure of a flower, of a typical flower, typical angiospermic flower, which has the essential and the non-essential whorls. So we go with the petals, which is the colorful part. Then you have the sepal, which is the green, which is in green color, and it is the protective part of the plant, which is when it is in bud condition, it is inside the sepals that the bud is protected, that the soft parts of the bud is protected. Then you have the filament and the anther, which comes under androecium. And the last one, the essential part of the flower, which is called ovary, style, and stigma, that comes under the gynoecium. So, anther and filament making up the androecium, and the um, ovary, style, and stigma making up the gynoecium part of the flower. So, here we go. The pre-fertilization events. In this portion, you will be learning about the stamen, the microspore, the important terms what we have in this. Let's see one by one. The typical stamen. The typical stamen, here we have the anther and the filament. So this is the filament, this is the filament, and this is the anther. These are the two parts that make up the stamen. Now, what are the important words that how you describe a stamen? Is it is bilobed. That is, this anther is bilobed. You can see two lobes there, one and two behind, and each one of those is dithecous. So when you see, this is one and this is one. The bilobed part, this is one lobe and this is the other lobe. Each of the lobes has two more theca. So the words, the important ones, what we have over here is bilobed and dithecous. Dithecous meaning, di meaning two. So you have two theca there, two theca, two lobes, bilobed. So when you have each of these lobes, inside each lobe, you will have two compartments. Each compartment is called theca. So totally you will have 1, 2, 3, and 4. These are the four, the important words that describe an anther. Moving ahead, the anther is four-sided, so it becomes tetragonal in structure, and all these would be having, of course, would be having the scientific term behind. It's called the microsporangia. In other words, microsporangia is pollen sac. So in lower classes, what we learn as pollen sac has a new name here, which is called microsporangia. So in the next paragraph itself, you have an important sentence telling the microsporangia further develop into pollen sacs. Let's go ahead with the structure of microsporangium. The structure of microsporangium is uh, circular, a typical one again. And the important part here is it has four layers. Let's see what are they. Epidermis, endothecium, middle layers, tapetum. So let's go one by one in order. The first one is epidermis. Write it down. The second one is endothecium. The third one is called the middle layers, and the last one and one of the important one, which is called tapetum. Now the outer three layers, that is, these three layers are protective in function, right? The outer layers protective in function, and this will be the innermost. This is the innermost layer, which happens to be the nourishing layer. What is it nourishing? It is nourishing the developing pollen grains. So that's about the structure of microsporangium. Now, when the anther is young, so you have two diagrams there related to the microsporangium. Uh, it would So the first one is about the young anther, and there you have an important word which is called the sporogenous tissue. What is the spore? Where do you find the sporogenous tissue? Sporogenous tissue is in the center of each microsporangium. Let's see how it is. So at the center, look at the diagram here. Each microsporangium, look at that, very beautifully arranged. Each microsporangium, this is one microsporangium, this is two, this is three, and this is four. Each of those have different layers. We have already learned what is epidermis, endothecium, middle layers, and the tapetum. Look at the arrow points. And each of these four microsporangium, in the center, it would be having the sporogenous tissue. So where is the sporogenous tissue here? This is one sporogenous tissue, this is two, this is three, and this is four. Now when you take an enlarged view of this portion, that is what is shown here. The enlarged portion would give you how, would tell you how these layers are arranged. So you have epidermis, endothecium, and middle layers forming the outer part, and tapetum is the innermost layer. And these microspore mother cells, they what are the mother cells actually? The mother cells undergo meiosis. Very important point. Mother cells are the cells that undergo meiosis. That is, these are the cells that divide meiotically to produce microspores. So when it is microspore mother cells, these are the mother cells that produce microspores. So that's regarding the young anther. Let's see the description once again. So we have the most important points there. We have the important points. Microsporogenesis. Microsporogenesis is, you can divide the word as microspore and genesis. Genesis means formation. So formation of microspores is called microsporogenesis. So let's once again repeat. You have the sporogenous tissue there, which is occupying the center of each microsporangium. Now, as the anther develops, the cells of sporogenous tissue undergo meiotic divisions to form microspore tetrad. So 1, 2, 3, and 4 together, they form one unit, which is called the microspore tetrad. And then they separate. They separate as microspores. What would be the ploidy? Look at the important question there. Ploidy of the what would be the ploidy of the cells of tetrad? It would be N, that is haploid, always, because these are the cells, these are the gamete cells. So gametic cells after meiosis, since you have a clue there, meiotic division forming the tetrad, that means the ploidy of tetrad would be haploid. That's it regarding the microsporogenesis. Let's move to the next part. We have already described the diagram here. Let's go ahead with the matured anther. Very important diagram. So these are the microsporangia. This is 1, 2, 3, and 4. Inside these microsporangia, what is happening is these microspore cells divide. And after division, what is happening there? They divide to form these tetrads. So if you look closely, you would find 4 cells attached together. So these 4 cells together, you call it as the tetrad. And when it is matured, they start separating into pollen grains. So this is an enlarged view of microsporangium, matured uh, anther, which is ready for dehiscence, meaning the microspora, the pollen sacs have it bursts out to give to release the pollen grains. So that's regarding the diagrams. Now, if you go by the explanation of this entire process, let's see how the process is developing. Here we have the microspore tetrad. Now, each uh, pollen, the microspore, the sporogenous tissue, what we have in the center, it is giving rise to the microspore mother cell. Microspore mother cell or the pollen mother cell divides meiotically to produce microspores, and this formation of microspores is called microsporogenesis. So very important word there. You can have the sentence there. The process of formation of microspores from pollen mother cell is called microsporogenesis. And what is the division that takes place there? What is the cell division? It is meiotic. Now, since it is meiosis, how many cells would be formed from one microspore? From one microspore mother cell, 4 microspores would be formed. So cluster of 4 cells which is in the form of microspore tetrad. Very simple logic. Each cell divides meiotically to form 4 daughter cells, and these, what are the 4 daughter cells called? The 4 daughter cells are called microspores. Which are the cells developing into microspores? They are microspore mother cells. Now, how does the anther release the microspores? What happens is, as they mature, they dehydrate. That is, they lose water and become more and more dry. When it becomes dry, the microspores develop into pollen grains. They dissociate from each other. That is, they get separated. And how many pollen grains would be formed inside these inside each of these microsporangia? Several thousands. Now here several thousands is given in your textbook, but when it comes in a question, say for example, if you have a question telling in a microsporangium, in an anther, each microsporangium is having 100 microspore mother cells, how many microspores would be formed? So typically, if it's a typical microspore and if it's a typical anther, it would be having 1, 2, 3, and 4. So each one having 100, meaning 400 microspore mother cells. So how many microspores would be formed? It would be 1600. That is 400 into 4. So this is how the calculation goes for microspores and microsporangium, microspores, microspore mother cells, and the microsporangium. So you need to have a count on what this several thousands mean. Coming to the structure of pollen grains, you have a diagram which describes the scanning electron microscopic images of different pollen grains of different flowers. So you have one, two, three describing how the outermost layers of pollen grain look like in different flowers. So they come in different shapes, different sizes, and different structures, different designs they have on the pollen grains. When you have to describe the pollen grains are made up of two layers. You have the outermost layer, which is called the exine, and the inner layer, which is called the intine. These are the two layers what we have of on the pollen grain. Now, what is exine known for? Exine is known for sporopollenin, which is one of the most resistant organic materials known. It can resist high temperatures, it can resist strong acids, it can resist any kind of atmospheric condition, so that it doesn't get destroyed over and over. When over the years, it never gets destroyed. And there is no it is said that there is no enzyme that has the capability to destroy this sporopollenin. Now, when you have an outer layer which is so thick and which is made up of such an hard outer layer, hard substance called sporopollenin, how does the male gamete come outside, right? So there you have a very um, important term again, which is known as the germ pores. These pollen grains has germ pores, which are apertures meaning small holes, pores. There are these pores. Uh, uh, since they are holes, they don't have the sporopollenin as the covering, and it is through these it is through these holes or these pores um, the pollen tubes grow. So that's regarding the exine. Moving ahead with the intine. So this intine, the inner wall, it's made up of cellulose and pectin. So this is one important term. Exine is made up of sporopollenin and intine is made up of cellulose and pectin. Now the cytoplasm is uh, surrounded by the plasma membrane as usual, and the pollen grain, when it matures, it has two kinds of cells. What are those two kinds of cells? You have the vegetative cell and the generative cell. So which is the vegetative cell? The larger one is the vegetative cell and the smaller one is the generative cell. Now, why is it big? It is big because it has the food in it, and it has uh, a nucleus there. The generative cell is typically smaller than the vegetative one. Now, this um, if when you have a typical angiospermic life, uh, uh, the lifetime of a plant, the pollen grains are shed at this two-celled stage. There are, however, there are remaining, I mean, there are species of angiosperms where the generative nucleus would be dividing into two, giving rise to two male gametes. So, uh, what is the cell division that is taking place in generative cell? That is mitosis. Mitosis. So when generative cells divide into two, you will have one vegetative cell, you will have two male gametes. So three-celled stage it becomes. Male gametes are again cells itself. So some shed the pollen grains in this three-celled stage, Others, they shed the pollen grains in two-celled stage where you have one vegetative cell and one generative cell. Let's move ahead with the pollen grains. When you come to pollen grains, there are people who have uh, allergic reactions towards these pollen grains. So there you have mention of Parthenium, which is it's also very commonly known as carrot grass, and it is said to it reached India as a contaminant along with wheat, it's called. It said so. It is important. Came through wheat, and it is one of the reason that causes allergy. Let's move ahead. Now, what are the uses of these pollen grains? That is what is mentioned here. The pollen grains rich in nutrients. There are countries which uses these pollen grains as tablets and food supplements, and it is also used as syrups. It is used as it is good for athletes and racehorses as it is extremely rich in different nutrients. It's nutrient rich pollen grains, right? So you have a diagram which tells you how the pollen grains are used, different types of pollen products, what they have. The next paragraph tells you about how long the pollen grains can survive, or how long can the how viable the pollen grains are. So uh, different plants have different viability when it comes to, you have different examples there. You have wheat, you have rose, leguminosae refers to the legumes, solanaceae refers to potato and tomato, the examples. These are the family names. So when you have the cereals like rice, and it loses its viability within 30 minutes. And for roses and legumes and the solanum, the solanaceae, it is viable for different months. There you have, next you have how to store the pollen grains. It is in liquid nitrogen. You will have reference to cryopreservation. Cryopreservation, which is preserved in liquid nitrogen. Look at the temperature, that is minus 196 degrees Celsius, and it is used in crop breeding programs so that the pollen grains can be used later for breeding purposes, and it can be stored at such low temperature without without getting destroyed. Let's slowly move ahead with the gynoecium part. So we have the pistil, the megasporangium, and the embryo sac. Now, when it comes to gynoecium, it is a female reproductive part of the flower. When it has single pistil or single carpel, it is called monocarpellary. More than one pistil, multicarpellary. When the carpels are fused, you call it as syncarpus. When they are free, it is called apocarpus. Pistil is another word we use for carpel. Now, each carpel is made up of three parts, which is called stigma, style, and ovary. Now, let's see one by one, where do you find the stigma, where do you find this ovary. The stigma is the landing platform for pollen grains, that is like a helicopter lands on the helipad. The helipad is the stigma. The helicopter can be imagined as a pollen grain. So it will fly around, and it may come, it may reach a stigma through wind, it may reach stigma through the insects, it may come through bats or whatever the agent of pollination is. Finally, the pollen grains, in order to grow the pollen tube, it has to land on the stigma, or the pollen grains that land on the stigma uh, are the ones that can grow the pollen tube. Now, the style, the style is the tube that extends from the stigma to the ovary. Ovary is the basal part which is bulged. Each ovary has locules. Locules meaning different compartments, and the placenta, right? Going back to 11th standard, they have the placenta. It is the attachment of ovule to the ovary. So that's regarding the basic things regarding. Now you have another set of diagrams here, again very important. This is a typical, if you take a hibiscus flower, you will have the stigma there, this is the style, and this is the ovary. The part on which the ovary is attached or ovary starts, it's called the thalamus. So there's, you have the thalamus, on over the thalamus you have the ovary, then the tube-like structure which is style, and the stigma. Now you have the diagrams there, again very important. The second diagram tells you the multicarpillary and syncarpus of papaver. That's papaver. You have reference to this papaver in later chapters. The third one, this one would show you multicarpillary, that is many carpels, and they are apocarpus, meaning they are free. So this is fused, and this is free. That's about mycelia. And this is the structure of anatropous ovule. Anatropous meaning, you can see it's like inverted. Anatropous ovule is typical diagram what you have for drawing, for sketching the diagram. Very important diagram as such, where you have the hilum, the funicle, micropyle opening. Micropyle are two poles you have, that is the where you have the micropyle, it is called the micropylar pole. The opposite pole is known as the chalazal pole. Within the ovule, you have the embryo sac. Then you have the nucellus, which happens to be the nourishing layer. The layers, like you have middle layers, here we call it as outer integument and inner integument. So these are the different parts you will have for labeling a typical anatropous ovule. So when you go by the number of seeds, what you have when you cut a fruit, that is how you count the number of ovules, because ovule is the structure in a flower that develops into seed. So it may be one in, like in mango, paddy, or uh, wheat. And when you cut a papaya or a watermelon, you will find many seeds. That is, the flower will have many ovules, as simple as that. So this will tell you what a funicle is. This will tell you what hilum is. Hilum is actually the uh, part which joins the stalk of the ovule, and the body of the ovule. That region is called hilum. Then you have two protective envelopes, like I said, two protective layers. It's called the integuments. And the two poles, which is called the micropylar pole and the chilazal pole. Nucellus is, as I said, it has food material, so that is nourishing in um, the nourishing layer, and it is within this nucellus. The within this nucellus, you have the embryo sac or the female gametophyte. What is a gametophyte? Gametophyte are the structures inside which the gametes are formed. So since it is called a female gametophyte, the female gamete cells are formed within this female gametophyte. So what is a female gametophyte? It is called the embryo sac. So when you have understood the structure, let's move to the process of my megasporogenesis. So let's compare it with microsporogenesis. You have microsporogenesis, the formation of microspores. Here you have megasporogenesis, that means it is the formation of megaspores. How are microspores formed? They are formed from microspore mother cells. How are megaspores formed? It is formed from megaspore mother cells. So megaspore mother cell divides meiotically to produce megaspores. There is one very distinctive difference between megasporogenesis and microsporogenesis. Let's see one by one. A single megaspore mother cell divides meiotically, of course, to produce the megaspores. So what is the difference here? Let's see. Ovules, um, what happens there is one cell starts differentiating, right? Out of all these cells, one cell begins to differentiate. That one cell is called the megaspore mother cell. Now it starts dividing one by one. So this is the dyad, that is, it's divided into two first, then it divides into four more. That's called the megaspore tetrad. Now, what happens there is the further division. You have this diagram B and C, very important diagram. A large megaspore mother cell, what we said now, it's dividing into dyad and then a tetrad of megaspores. Now, this diagram tells you this is an embryo sac. The embryo sac has two cells there. It is dividing the nucleus of inside this embryo sac is dividing mitotically now. So first mitotic division 2, second mitotic division 4, and the third mitotic division there. So 2, 4, and 8 nucleate stages within the embryo sac. So now what's happening? Let's see in detail. The megaspore mother cell undergoes meiotic division. It results in the formation of 4 megaspores. Now here, the major distinctive difference between the microsporogenesis and megasporogenesis is that all the 4 microspores are functional. However, with respect to megaspores, 3 degenerate. That is, 1 megaspore mother cell would give rise to 1 megaspore. Only one is functional, the other 3 degenerates. So please don't take it wrong. Every meiosis will undergo, every microspore mother cell, megaspore mother cell would undergo meiotic division and it gives rise to 4 megaspores. However, out of these spores, the 3 degenerate. And only one. So which are the 3? Let's go back to the diagram. Look at this. So we had the megaspore mother cell there, which forms a dyad first, and the tetrad. Now 3 degenerate and only one would function, and that one is shown here, right? That's how it is. I repeat, we have 4 megaspores here, 1, 2, 3, and 4, out of which 3 degenerate, and this one is developing. That is where the mitotic division would begin. So let's go forward with the process. So this paragraph will tell you how how the nucleus inside this megaspore would divide. And what is the division there? It is mitotic, very important. So first mitotic division, it is forming two nucleate stage. Second and third, it forms 4 and 8 nucleate stages. Now, after the eight nuclear, when it reaches the 8 nucleate stage, the cell walls develop and it forms a typical embryo sac. A typical embryo sac will be formed when the megaspore would be reaching the 8 nucleate stage. That is when you call the megaspore as the female gametophyte or the embryo sac. Now here, next what happens is 6 of these 8 nuclei would be surrounded by cell walls and it is properly developed into cells. The remaining 2 nuclei are called the polar nuclei, which is uh, developed or which is situated below the egg apparatus. Right? So let's go to the diagram to understand how it is. This is how it goes. So you have at the chalazal end, you have the cells called antipodals. Then you have the polar nuclei. Then you have the egg cell near the micropylar end always. So wherever you have the pore, micropylar pore or the micropylar end, that is where you have the egg cell. That is very important. So if you draw micropyle towards this end, the egg cell should be here. If you draw it here, this is the portion where you will have the egg cell. Then you have the filiform apparatus which guides. So these finger-like projections are called the filiform apparatus. You have the two synergids and the egg cell. Together it is called the egg apparatus, which is seen at the micropylar end. So that is regarding the diagram, which is this diagram being very important for drawing, for sketching, and for describing the typical embryo sac. So the arrangement of cells, this is how it is. The egg apparatus having two synergids and one egg cell, which end has the egg apparatus. It is the micropylar end. Then you have the antipodals towards the chalazal end, and this is called the 8 nucleus. It has the embryo sac would be having 8 nucleus, but it is only 7 celled since you have a large central cell with 2 nuclei. So 2 polar nuclei, which is called the central cell. So how many cells in total? 7 cells. How many nucleus in total? 8 nucleus.